talk a little bit about evolution and membranes and amino acids, and hopefully you'll get something out of it. So I'm kind of looking at the origin of life from an evolutionary perspective. And so we know a little bit about what life looks like today. We know a little bit about what the Earth would have looked like, before life existed. And so we're kind of interested in how we get from one to the other. And so the kind of guiding principle that we're going to use and take advantage of, that a lot of people here are doing as well, is natural selection, which stated in a very simple way is that things that happen to work are more likely to stick around. And so I'm going to focus on proteins a fair amount because they're something that's very important to life as we know it. So about 20% of you is made of proteins. And they're one that's very problematic from an origin of life perspective because they're very difficult to make. The way that modern proteins are synthesized uses enzymes, which are already made of proteins, so you have kind of a chicken and egg problem to begin with, and some sort of informational polymer to, figure out how you want to stick your proteins together. But there are other problems too. So even if you have a really good assembly mechanism, if amino acids wanted to just spontaneously stick together, you still need to have them around in reasonably high quantities, which is something that I'm not going to address too much directly today. It's not that evolution would ever be like, hey, if we had amino acids, we could make these proteins, which would be super useful, so we'll make amino acids. It's not how it works. It uses things that are already around. And so kind of defining life is a little bit tricky, especially, at what point you move from non-life to life. And so the kind of characteristic that I'm gonna focus on is that life is out of equilibrium. It's different from its surroundings. And so generally it uses some sort of energy source to increase organization and order. And so kind of with this in mind, a couple things that we really want are some sort of metabolism to make useful molecules, and some sort of enclosure to keep your useful molecules together, right? So it's not enough just having all of the pieces. Like if you dissolve a human in a bathtub, it's no longer alive, even though all the molecules are still there. So I think this is a very important component. And then you want some sort of robustness and replication. You want something to stick around for long enough that it can make more of itself. But I'm not going to focus on that too much today. I'm really going to focus on kind of the enclosures aspect in addition to the amino acids and proteins. And so we know a couple things about prebiotic membranes. We have some plausible ones, at least, that could have existed. And these are mainly kind of fatty acid vesicles, but they have some problems. We know that they're not perfect for primitive enclosures. And their kind of biggest problem is that you need really high fatty acid concentrations in order to get these things to form and to stay formed. So even if you make a vesicle out of fatty acids, if you move it to a place where there's low kind of dissolved fatty acid concentration, it falls apart. And then they're generally incompatible with divalent salts, which we probably had around too. And I think one of the things that is a really good idea, and Roy Black has really been kind of championing this idea, is that inclusions into vesicles can change their properties in very dramatic ways and might be very helpful for getting something that is prebiotically relevant and would actually work. And so I'm going to look a little bit at how membranes systems exist in modern life. And so they're largely made of surfactants. It's one of the main components. And they're generally, in very simple terms, molecules that have one side that likes interacting with water and one side that really doesn't. And so they kind of form these bilayered membranes that separate, the outside of your cell from the inside, keep some things out, keep some things in. But that's not the only thing that is, in our modern membranes. There's a lot of proteins. And these proteins perform a lot of very important functions, to life. And so, they keep everything structured. They control transport of material inside and out of cells. They have a pretty large role in signaling, whether it's getting a cell to swim around or getting me to stand up here and talk at you. And maybe surprisingly to a lot of people, they have a really big role in energy metabolism. So a lot of the proteins that are involved in photosynthesis and oxidative phosphorylation are actually membrane proteins. And so looking back at the relationship between amino acids and proteins, kind of the local amino acid composition of a protein controls its function in a lot of ways. And so a very simple example of this is just a membrane protein. I have a channel type thing drawn here very crudely. You generally have these kind of hydrophobic amino acids in spots where it's interacting with the kind of hydrophobic part of the membrane, and so it's kind of stable in the membrane and wants to stay there. And then you have this hydrophilic stuff that forms your channel. And so I think an important question to ask is, do you actually need, or can the amino acids perform some of the roles that proteins do today without being assembled into proteins? And so some of the techniques that I use to investigate kind of similar questions are surface-sensitive techniques. So I'll look at a single monolayer of surfactant on a water surface. This is nice because it's a very controlled system, even if it's a little less relevant. So you get to kind of scrape these barriers across the water surface, and you can control the relative surface concentration of your surfactants, and you get thermodynamic information by measuring surface tension as you change the surface concentration. You can use this in conjunction with other techniques like Brewster angle microscopy, which is really neat because you get to basically take pictures of single molecule thick films at water surfaces. And so kind of an example of some of the interactions between a membrane type system and a amino acid. It's not the most prebiotically relevant. I don't think people thought phenylalanine was around prebiotically, and DPPC certainly was not. It's a big complex molecule that all of you are full of. But at any rate, I think it's a good example of kind of a very strong perturbation by an inclusion into a membrane. And so phenylalanine incorporates into this monolayer of DPPC, and right away between these two Brewster angle microscopy pictures, you can see there's a huge change in morphology. It has kind of a condensing effect on some regions of the film, and alters the stability by interacting with DPPC in favorable ways. And kind of an interesting point about all of this is that different amino acids behave very differently, even if they look very similar. So it's not always going to be easy to predict how things are going to behave. So up here I just have like the solubilities of these different compounds, and they're all across the board. And it actually turns out the key to understanding the differences in this system is the clustering of these amino acids. So these two like to cluster with themselves once they're inside the membrane. They don't cluster in solution, but in the membrane they do. And this really controls their kind of membrane altering behavior. And I don't know how general this is, but I think it's a really neat effect, particularly because if you have aggregation, a lot of the times that's chirality dependent, and so it gives you something that is potentially selective for chirality. And so I didn't talk about the most prebiotically relevant system, but I think that it's important for illustrating a point. And then the other issue is that the types of membrane alterations that you want by your specific inclusions are going to vary dramatically based on the composition of whatever type of membrane you're talking about. So if you have something that's like a fatty acid membrane that's already very permeable, but it's not very stable, you're probably going to preferentially select inclusions that are going to increase the stability. But if you have a different type of membrane, like modern phospholipid membranes are very stable, but they're not permeable, and so those types of membrane systems, you're going to select for things that are going to increase permeability. So you have to remember that when you're looking for inclusions to perturb something, it depends what properties your initial thing has. And then the other point is that for any prebiotic scenario, we're not going to have a single isolated compound or a mixture of two. It's going to be something that's much more complex. and it's going to have its own set of criteria. So it's something that's very hard to investigate, but I think worth doing. And kind of likewise, having a variety of amino acids is going to let you kind of selectively modify different characteristics depending on which amino acids want to incorporate. And then kind of the final point that I wanted to make is that I think that membrane systems are very promising for as far as making the transition from amino acids to proteins, just because the amino acids kind of have a role to play in the first place, but also because you have a couple of advantages. So if you are getting something that's kind of intercalating into a membrane in the first place, you're inherently increasing the concentration. You're moving from a three-dimensional space into a two-dimensional one. The clustering allows the organization, like I said before, and potentially is chirally selective, which is another interesting topic. And it's also a very different chemical environment. So normally making peptide bonds is unfavorable in water. And so the membrane is kind of a hydrophobic environment that might help. We've actually seen that water interfaces can be favorable for amino acid or for peptide bond formation in the past. But I think the most interesting part of this is that it doesn't necessarily require any sort of informational polymer to assemble because you're already kind of intercalating into the membrane and clustering in a way that's useful. And so that's kind of your selection for making a useful protein in the first place. And so with that, I'd like to acknowledge my group and some of the collaborators that I've worked with and a little bit of the research that I showed and a little bit of funding.
All right, any questions? Have you experimented with the different amino acids for membrane stability? Because you pointed out the theoretical stability. So building upon that work, because you have a whole suite to work from.
Yeah, it's kind of an interesting thing because in a lot of ways it depends what type of stability you want. So, really I should probably be moving a little bit more into vesicle type of systems. Where you can see what it takes to make your vesicle fall apart, resistance to salt and things like that. Stability is kind of confusing for looking between two-dimensional and kind of three-dimensional vesicle systems.
Do you think some would actually provide stability to divalent cations, which is a big problem with fatty acids?
I think that there is almost definitely something that will. Whether it's an amino acid or not, I think is questionable. But
the magic of phosphocholine, right?
Yeah.
I was just wondering, it's not a amino acid, but incorporation of cholesterol into the membranes.
Yeah.
Has anybody tried that and saw how that affects their stability? Because I know that's how cells regulate it now.
With modern cell membranes, certainly cholesterol has a huge role to play. I'm not sure it's going to be the most useful one for fatty acids because generally what it does in kind of phospholipid type membranes is it makes them a little bit more fluid and kind of more permeable, which is something that's not really an issue for fatty acids to begin with. So it's almost like you're trying to fix the wrong problem if you're using a molecule like that in a fatty acid
the opposite
system.
Okay.
Yeah.
All right, thank you very much.